Hello everyone. Today I'm gonna share with our recent study about using deep learning method for identifying post-translational modification sites, specifically lactylation sites. First, let's go through the post-translational modifications. Technically, they are covalent processing events that change the properties of amino acid or a protein. There are two important aspects in the study of PTM are identification of the site of the modification and understanding of how the change in that site affects the functional consequences for the protein. Those consequences include activation of protein, changes in its localization or stability, or changes in protein-protein interactions. Specifically, we focused on a newly discovered PTM named lactylation. It has been demonstrated to be involved in different biological processes. For instance, Yu and their colleagues found that histone lactylation is elevated in tumor and associated with poor prognosis of ocular melanoma. Also, recent studies have found that lactic acid in the blood of patients with sepsis is directly proportional to the content of HMGB1. So we could conclude that identifying lactylation sites is a very important step for understanding of different kinds of biological processes. Basically, we often use MS-based sequencing technique to mapping PTMs. As you can see here, there are a few steps such as sample preparation, digestion, MS processing, database search, and signal integration. Apparently, it is tricky to do compared to computational method. So, inspired of this, we designed a deep learning model to identify lactylation sites. This is our workflow, including benchmark dataset construction, feature extraction, model training, model performance evaluation, and web server build. First, benchmark dataset construction. One important thing we need to do is mapping the modified protein and modified position to the UniProt database. After doing that, we could get protein sequences. Basically, there are some redundant information involved in protein sequences. So we often use CD-HIT program to remove sequence similarity. Finally, the benchmark dataset, which contains positive and negative samples, was constructed. Next, there are two main steps, including feature extraction and model training. Since we designed a deep learning architecture, so the processes of feature extraction and model training were combined. Our algorithm consists of 4 parts, including embedding layer, CNN layer, R layer, and attention layer. Next, we used ROC curve and AUC value to assess the model's performance. As you can see here, our proposed model can produce satisfied performance on training data as well as independent data. Both of them are higher than 0.9 in terms of AUC value. In the end, we built an online service for easy use. You can directly input query sequences into box, and also you can upload file to predict. But note that please input FASTA format file. After clicking on Submit button, you could see a result table, which records query sequences ID, modified position, modified residue, and prediction probability. That's all. Thank you for your attention.